Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash. Hello, everyone, and welcome back to the Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, how are you doing today? I feel like usually I just talk. Mike, how are you doing?
I love hearing you speak, Molly, and I'm doing fantastic. I'm great. I'm energized. I'm drinking caffeine. We got not a Fresca today. We got some Zoa. If anyone's a Costco fan, shout out to Zoa. Not sponsored. And we're talking about something that's very related to what we talked about last episode. Because and this is a topic that, especially when we teach, again, we talk about it so much, and we teach so many classes between you and I. This is probably one of the topics today is probably one of my favorites because I love talking about stuff that people overlook, right? Especially when it comes to the field of metabolism, right? Because if you clicked on today's episode, you got to know what we're talking about. Because last episode, we talked about glycolysis. We talked about the pathways. We talked about, we really, you and me, Molly, we went in depth on glycolysis. It was an action-packed
episode.
And so we went through all the steps of glycolysis, going from glucose, ending on pyruvate, what was produced. And now this episode, we're going to be talking about the next steps of glycolysis. What happens after that point? It's in the context of aerobic metabolism. Sure. Yes. At the end of glycolysis, you actually have a few different options to do, but we're going to go through the aerobic metabolism pathway to produce energy. And where we're going to start off today, we're going to be talking about two things today. Where we're going to be starting off is one of my favorite things to talk about, and then we're going to be going on to one of Molly's favorite things to talk about. And so we're going to be talking about the Pyruvate Dehydrogenase Complex and the TCA cycle today. Molly, I love these things. What are your thoughts? When you teach these things, what are the vibes when you teach them?
I love all of metabolism because most students hate all of metabolism. And we've talked about this. We've brought this up again and again. If you are a regular listener, you've heard us say this, but my absolute favorite things to teach, like the things that get me animated and bubbly and excited, are the things that I hated for most of my college life and into my actual MCAT experience. I hated these things because they were fundamentally misunderstood in my brain, right? I didn't learn them in a way that really made them click. And then I was frustrated because I am not a good person at memorizing. And so I felt really defeated by a lot of these topics. And metabolism was one that I just felt I'm never going to be able to memorize all these steps. I'm never going to remember. I learned the Krebs cycle or the TCA cycle, for example, which is something we'll talk about today. I learned it in OChem. I learned it in biochem. I learned it again for the MCAT, and I felt like I was relearning it each time. Same with glycolysis. Now I don't forget it because I understand it. And I love being able to pass that on to students because at least me, I don't know if you struggle with this too, but I know your pain points because I've been there and I hated this material too. Hopefully we can find a way to get you excited about this material. Maybe not quite as excited as we are yet, but maybe down the line, right, we can make you like this. So I love talking about metabolism. And ultimately, like you said, we're going to go through aerobic metabolism first. So glycolysis, we'll talk about the Pyruvate Dehydrogenase Complex and the TCA cycle in this video. The next video is actually going to be my favorite, which usually we tease it, but it's the last step of aerobic metabolism.
Well, I wonder what it might be. We'll leave it at that.
We'll see if you can guess. That one's actually my favorite. I'm very, very excited for it. And then we will cover some of those offshoot pathways after our next episode. So there are lots of...
We got plans.
We got plans, you guys. So stay tuned. But let's start with the PDC. You said that you love this
I love
stuff,
talking about the PDC.
so let's hear it. Mike.
Of course, Molly. The reason I love it is because, like I was saying before, I love talking about the stuff that people overlook and don't really think are that important. The first time, I think one of the first times when we talk about mixed inhibitors, everyone doesn't give mixed inhibitors any love. And I'm like, no, they're so important. I love them because they're so cool. The PDC is also a great one where a lot of times when I ask students, okay, what happens after glycolysis? Because everyone knows, this is something when you first metabolism, okay, glycolysis is the first step. Everyone's like, oh, after glycolysis, TCA cycle, Krebs cycle.
TCA cycle, yeah.
Right? And I'm like, there's a really important step in between them because we have to recall, we've got pyruvate. We ended glycolysis. We've got pyruvate that's in the cytoplasm. The TCA cycle, teaser, it's acetyl CoA that enters into the TCA cycle, but we need something in the middle there to turn that pyruvate into acetyl CoA. And that is where the pyruvate dehydrogenase complex comes in. And now, I want you to, before we even dive into, think about the name, right? Pyruvate dehydrogenase complex. Based on just the name, what do you think is happening?
Redox.
We love the redox reactions. The name
or
of the
reduction
enzyme,
or oxidation.
Everything's happening because they happen together. It's all happening. And so and this is where we were talking about last time, especially with a lot of the glycolysis enzymes, and we're going to see that a lot in especially the TCA enzymes. The names tell you so much about what's going on.
Yes.
Don't try to memorize, oh my gosh, what's happening here? Think of the name, and that tells you half the story, right? So the pyruvate dehydrogenase complex tells you there's a redox reaction occurring during this step. And as well, there's another aspect that's really important. It's a complex. So the PDC is not just one enzyme. So we're going to talk about that. So first, let's give an overview. Where does the PDC happen? Now, we talked about last episode, glycolysis happens in the cytoplasm. That's the hint, and we, again, bringing up some evolutionary biology, which I know that you love, Molly, because it happens in the cytoplasm, very old mechanism, very like ubiquitous, really with all cells can do glycolysis. But now, the PDC is something that's much more specialized in aerobic metabolism. And because it's specialized, it happens in the mitochondria. Right. And so the PDC happens in the mitochondrial matrix because there's kind of two compartments of the mitochondria. There's the matrix, which is kind of the inner part, and there's the intramembrane space that's between the outer and inner membrane. And so we essentially have to get the pyruvate into the mitochondrial matrix before we begin. Right. So this is where the PDC occurs. And now, as well, we have to think about what's overall happening. It's happening in the matrix, but we have these pyruvates. Now, we recall something I mentioned last episode. Again, I'm going to keep referring to that because it's so related, right? Pyruvates each have 3 carbons, 3 carbons each. But if we think about what the goal is, pyruvates go to acetyl CoA, right? And acetyl CoA, Molly, might have how many carbons each?
2.
Acetyl CoA have only 2 carbons each. But wait a minute. If I started with 3 carbon pyruvates and now I'm ending with 2 carbon acetyl CoAs, there's something missing here.
No, it just disappeared actually.
It disappeared.
Thin air.
It just disappeared.
Just
Right?
poof,
We actually
gone.
violated conservation of mass. We broke some laws.
Yes,
Absolutely
exactly.
not. Of course. Right?
It's a magic trick.
It's a magic-
Mike, what do you think is going on? Duh.
No, that's where we are taking the role of the PDC. We know a redox reaction is happening, but we also see we are removing a carbon from that pyruvate. This is the first time that we're actually removing carbons, because recall, in glycolysis, no carbons were removed. We started with 6 carbons, we ended with 6 carbons, and 2 3-carbon molecules. Now, we go from pyruvate, which is 2 3-carbon molecules, to 2 acetyl CoAs, which are 2 2-carbon molecules. So 2 carbons are removed and turned into CO2, right? And so whenever we remove carbons from from those original carbons from glucose, it becomes CO2. Recall like that. And this is kind of when you talk about the overview, right, that we talked about last episode. We're turning glucose into carbon dioxide. This is an oxidation, right? And so we're essentially oxidizing it. But wait a minute, based on what we just talked about, is there any surprise that oxidation is happening in this reaction?
No, because of the name of the enzyme.
Dehydrogenase. We already knew a redox reaction was occurring. But wait a minute. If we're oxidizing those carbons, what must also be occurring at the same time?
Reduction.
Reduction's also
I'm
gotta
getting
be happening.
100% so far today. I'm
You, Molly,
not to brag, but
you are rocking it. Like, have you seen this before? I don't know.
But I've studied it once or twice.
But yeah, so we know those carbons are being oxidized as a part of the PDC. But therefore, whenever you have oxidation, something has to be reduced. And most of the time in aerobic metabolism, when you see reduction, NAD is involved. NAD plus is involved. And when we're reducing, those are gaining electrons. So NAD plus is gaining electrons, forming NADH. Now, the NADHs are those really important high energy electron carriers that we're going to be talking about, become very relevant when we talk about that last step that Molly loves. Hint, hint for our next episode. But this is something that's really forgotten about in PDC, because people always kind of think about that step like, oh, the step doesn't really matter. It's like a linker step. No, energy is produced or energy is extracted from that pyruvate that's going to be used in the electron transport chain. Ah, darn, I said it. But like I was trying to tease.
Spoilers.
Darn.
My gosh.
But yeah,
Yeah.
so both CO2 and NADH are produced in this step. So two pyruvates are converted to acetyl-CoA, and in that process, 2 CO2 are produced and 2 NADH are produced. Okay.
Yes.
So this, like the PDC is one that's important to know, but that's really the kind of key about it, right? Knowing the conversion, like where do you start with, where do you end with, like what do you end with, and the process to get there in terms of what reactions occurred. Like I was saying before, it's a complex. So it's not just one enzyme, right? It's actually 3 enzymes all together. I don't think I've ever seen an MCAT question ask about the names of the other 2 enzymes. There's one enzyme that's very important, which is pyruvate dehydrogenase, which is actually the first one. But the other 2, like, I don't think I've ever seen a question ask about them. But because it's a whole complex, there's a lot of moving parts. And because there's so many enzymes, there's also key cofactors involved. Now, recall when we talked about cofactors, I think this was a few episodes ago when we talked about like enzymes, like cofactors are those things that, the enzyme needs to be active to do its job, right? It's an apoenzyme until it adds the cofactor. Now it's a whole enzyme or a hollow enzyme. Some key cofactors with the pyruvate dehydrogenase complex are 2, which we're going to talk about some really cool applications of them, are thiamine pyrophosphate or vitamin B1. Thiamine is just a fancy name for vitamin B1, so eat your vitamins, as well as lipoic acid. Like lipoic acid is another very important cofactor for the pyruvate dehydrogenase complex. So again, the MCAT will expect you to be familiar that there are cofactors involved with the PDC. And whenever you get and I don't know about you, Molly, but whenever I hear cofactors in aerobic metabolism, it's usually a question about the PDC. And so these are kind of the 2 key cofactors to know about: thiamine pyrophosphate, or which is just vitamin B1, and lipoic acid for the PDC.
Yeah. I always tell students, PDC, like you said, it's very overlooked, understudied. It's seen as just one reaction, one step, and that's it. But like you said, it's not. It's okay to study as one reaction. Like you're totally free to do that. We're going to treat it like that on the MCAT. But there are a lot of different enzymes that are part of this that are all doing this weird little handoff of doing each their own individual component of this reaction that's producing NADH, that's producing CO2 and acetyl-CoA. As far as the cofactors, I always just tell students, if you get a question about cofactors with relation to metabolism, it's PDC, right? That's where we're going to talk about cofactors the most, right? Of course, NAD plus, FAD, we're going to see those elsewhere, but TPP and lipoic acid are really important. And it's really cool because do you need to make this the number one, focus point of your studies? No. But there are lots of really interesting but terrible consequences of messing with these cofactors. So I like this because, again, I didn't love trying to deal with that material earlier when I was trying to prepare. So this I just think is cool. A lot of us have heard about arsenic, right? Arsenic
scary.
Poisoning. Yeah. I think it's pretty commonly known poison. And it is very, very dangerous, right? Extremely. It's going to create basically system-wide organ failure, vomiting, even garlic breath, which is interesting.
Interesting mechanism.
I don't know the reason behind that, but I've heard that a couple places. But the actual biological mechanism for arsenic poisoning is actually inhibition of lipoic acid. So the actual physiological problem that arsenic poisoning creates is that it will shut down lipoic acid, which is required by the pyruvate dehydrogenase complex. So what we're going to see is we can't take pyruvate and make acetyl-CoA, which means, can we do TCA cycle? Can we do ETC with that, with those, original
molecules?
Yeah.
Absolutely
with those
not.
glucose byproducts.
No, right?
No, you can't, right? There are other pathways, which we'll talk about a little bit later, that you can make acetyl-CoA from other molecules like fats. or from like amino acids. There's a couple different molecules that can create it to make energy, but you can't do it from glucose because glucose makes pyruvate through glycolysis. If you can't do PDC, you're probably going to be leaning on glucose a lot, which means you've got a lot of anaerobic metabolism going on. Specifically, it's glucose and fermentation. You might have oxygen, but because you can't do pyruvate to acetyl CoA because lipoic acid is not working, you can't go into the TCA cycle with the metabolites of glucose. So you do a lot of fermentation, which means you've got a lot of lactic acid buildup, which, keep in mind, just takes the electrons from NADH and sticks them onto pyruvate to make lactic acid. That's going to build up and it's going to create lactic acidosis.
Right, which we've talked about with when it came to lungs and those sort of things. pH regulation. Oh my goodness, the callbacks. If you don't know what I'm talking about, go watch those episodes. We did one on like we had a whole series on homeostasis where we talked about the lungs, kidneys we talked about the kidneys, and endocrine system, how pH was involved. And so highly recommend going back on those episodes to see how the body deals with those changes in pH. But absolutely, we can see something like arsenic poisoning can absolutely impact the pH. And again, I love these applications where it might not be MCAT testable, like about arsenic poisoning, but you could definitely see a passage about that where they make that linkage in the passage. Which I think is super cool. Another great application would be the thiamine, because you kind of focus on the lipoic acid factor. Arsenic kind of inhibits that lipoic acid, doesn't involve it in PDC. Thiamine, that other cofactor, can also be inhibited, and a thiamine deficiency is known as Wernicke's or Korsakoff syndrome.
Not that you need to know that.
Not that you need the names, but this is a side effect of overconsumption of alcohol. So overconsumption of alcohol. Alcohol is something that's appeared more than once on this podcast. I swear it's not a trend on purpose, but it's just because it has so many systemic effects on the body. And so alcohol intake actually inhibits the uptake of vitamin B1, again, that really important vitamin, which is thiamine, for the PDC. But again, if I can't absorb vitamin B1 from my diet, I'm going to be lacking it. I'm going to be deficient in it. If I don't have any thiamine, I can't do PDC. If I can't do PDC, the exact same problems happen, where I can't make acetyl CoA from my pyruvate. Pyruvate it's going to be turned into lactate using that lactate dehydrogenase, because I have to get those NAD pluses back to keep glycolysis going, because that acetyl CoA is not producing any energy now because I'm not even producing it. And so the same things are going to happen, but this is a different mechanism entirely. Like, it's not arsenic poison, but this is because of an overconsumption of alcohol, which is, again, is this MCAT content? No. But is it a cool application to link how important the PDC is? Absolutely.
Yeah. And I feel like, especially when we have things that we feel we just don't care about, or things that we're like, this dumb little detail could show up, it's awful, make it fun, right? Go Google, like, why does lipoic acid matter in PDC? And you'll see results, right? Like, if you don't, then maybe it doesn't matter, right? Like, if there's no consequence of removing it, was it really that important? I don't know, right? That's oftentimes the MCAT is going to focus on things that are biologically very relevant. And messing with biological systems have a lot of consequences. In the realm of consequences and maybe turning things up and down, we do also want to talk about regulation. Definitely not as big of a focus as glycolysis. You'll notice none of the discussions in this episode are going to be quite as deep as glycolysis, because that really is the reaction that you need to know in the most depth, because it's connected to so many others. But for this particular reaction, regulating the activity of the pyruvate dehydrogenase complex, I actually think it's really, really cool to talk about. So the typical activators and inhibitors are pretty much exactly what you would expect. Like, they make sense. If you don't want, or if you have a ton of energy in the cell, you already have a ton of ATP, I don't want this thing going. Because as we alluded to last episode, when we talked about the irreversible steps of glycolysis that actually can be reversed, they just need a different enzyme to do it, right? If we wanted to go through gluconeogenesis, you could. Here, this pyruvate to acetyl CoA, you cannot go back.
No way.
Once you have done that, you are stuck as acetyl CoA. If you don't want to make ATP, you could use that acetyl CoA to make fatty acids and fats, lipids. You could store it, but you cannot make glucose from acetyl CoA. Super important. So because of that, this regulation is actually quite important. And again, if you have a lot of ATP, you're going to inhibit it. If you have a lot of NADH, which again can be used in the ETC to make ATP, you're going to be slowed down. If you have a lot of acetyl CoA, that's going to be slowed down, right? What do we call that when the product inhibits the enzyme that makes it?
Negative feedback. I was so ready. I was like, I knew you were gonna ask.
All right, you're 1 for 1 right now.
We're
I'm
winning.
gonna call on you again later.
We are up.
Yeah, these are all things that signal a high energy state. Either the cell already has a lot of ATP or it has a high capacity to make it. Things that would be activating ADP, right? That's saying that we don't have as much energy available. Again, we take ADP and we make ATP. So if we have a lot of ADP around, we probably don't have as much ATP. NAD plus, if we have a lot of sort of empty electron carriers, we might want to make more because that's related to energetics as well. And a lot of pyruvate, right? If we've got a ton of it, might as well use it. The one thing that I think is really cool though, and I always bring this up and I always make this a highlight, especially now that we've talked about enzymes, is actually how this complex can be regulated through phosphorylation. Because typically, and we mentioned it a couple episodes ago when we talked about enzymes and enzyme kinetics and different types of enzymes, right? Enzymes that phosphorylate, right? Which would be what?
Kinase and phosphorylase.
Yes. Enzymes that phosphorylate, right? And we'll talk about kinases because it's adding energy from ATP, or adding a phosphate from ATP. What does that normally do to the activity of an enzyme that's going to be phosphorylated?
Like generally on the MCAT, of course, generally, not always, of course, generally we see it activates things.
Yes. Yeah. Typically it's activating, and it kind of makes sense because if you want to activate an enzyme to go do something, you want to make sure that if it requires energy and down the line, you already have enough. Like you don't want to activate it in a low energy state. If you didn't have much ATP, like maybe that protein shouldn't be active, right? It depends on what the protein does though. And the reason why I like bringing this up is because in this case, right, because this complex is specifically designed to help commit energy dense molecules to creating ATP. If we have a ton of ATP laying around, this is going to do the opposite, right? Normally, if you have ATP laying around, you might as well use it for phosphorylation, go activate downstream enzymes, right? They go off, carry out their function. If they need ATP, we've got it. It's fine. In this case, if I have a bunch of ATP laying around, I want PDC off. Because if we have ATP, why would I want you to go make more? Again, I always think of it as taking money out of a bank. If I already have $100 in my pocket, do I really need another 100? If I already have $20,000 in a safe in my house, do I need to go get another $20,000? I don't. I wish.
Yeah.
I wish I had $20,000.
I mean, if I had 20, but why? I'll go get another 20. Let's make it 40.
I mean, who's paying, right? Like, why would you need? Why would
You don't
need
need more.
Why would you need $40,000 in cash if you're not going to spend it, right? Like,
body doesn't. I know.
right. You don't need. It would be better to save that as either a fat with acetyl CoA or as glycogen, right? Storing glucose. So ATP is going to be used by pyruvate dehydrogenase complex kinase, or PDC kinase, to turn the pyruvate dehydrogenase complex off. And if we want to keep it phosphorylated, right, we're going to make sure that phosphate stays on. If we do not have ATP anymore, right, and we may then need this to come back on, we're going to use PDC phosphorylase to chop off that phosphate on PDC using,
phosphatase. Cool.
Sorry, did I say phosphorylase?
Yeah, I was like, I heard lase, and I was like,
phosphatase.
Yes.
Thank you.
Yeah.
The naming,
I know.
is annoying, but yes, a phosphatase removes a phosphate. Thank you. Yes, and then we will have PDC active again.
Mhm.
So that is an extra layer of this regulation as well. We not only have molecules activating and inhibiting, but we also have enzymes that are helping us regulate. I like to bring it up just because, again, most of us think of activation as phosphorylation, but sometimes phosphorylation can deactivate. And it really depends on what the enzyme does, right? Do we want it active when we don't have energy, or do we want it active when we do have energy? That will pretty much tell you what we're doing.
And I think this is a cool point about the body in general. Like you can't imagine how many questions I get like during classes or during the free trial sessions that we host, where it's like, oh, a student asks like, is this always the case?
I love that question because I'm like, I will never say yes to that.
Yeah, never. Because like
I will never say yes.
If I say yes to, oh, is phosphorylation always activation? I'm sure if you look it up, you'll get a million situations where it's not. And so the body is really cool where we can't generalize most things. Like if we are able to generalize like something, that means we are really certain about that thing. But in science in general, like even when our data, we can't ever even say with 100% certainty that something occurs. Like we have a p-value for that reason. And so we can never like say, oh, phosphorylation always activates, right? Because we can see that it doesn't. Right. And so I think this is just important to keep in context when we think about like generalizing the sciences. A lot of times you can't for most things, which I think this is just another great example of you can't generalize, oh, phosphorylation means activation, because like you said, that isn't always the case.
Yes. Yeah. I love it. See, we have so many fun conversations with metabolism. It just sparks so much joy now in my heart.
And not pain at all.
Not pain anymore.
But you know what's another area that I know I had pain with? And because I learned it over and over and over again and never got it until I studied for the MCAT.
Never learned it well.
Was the TCA cycle, which is the next cycle that we're going to talk about, because this is one thing, and I'm sure most of the people listening, if you've taken the science classes, like you were saying before, we've learned this thing like probably multiple times. Like we learned it in high school. Most of us learned it in like grade 11, grade 12 biology, or I'd say that as a Canadian, maybe in your junior or senior year of high school.
There you go.
I'm like, oh, I have to think of like an American right now. But then also you learn about it in like cell bio. You learn about it in organic chemistry. You learn about it in biochemistry. Like you learn about this pathway so many times. Yeah, it always feels like we're just learning it one more time. It's like, oh, I forgot it now. I'm gonna memorize it, then forget it. Memorize it, then forget it. But like we were saying before, this is not a memorization exam. The MCAT's not going to ask questions about the TCA cycle where your goal is just to memorize the intermediates. It's important to understand what is happening during this time. Like the enzyme names are going to be really, really important for this process, to understanding, okay, what's the point of the TCA cycle and actually the really big picture. And so kind of let's start from like I think it's always great to start with the big picture, to kind of think about, okay, where did we just come from, right? We just came from the PDC. We just converted that pyruvate to the acetyl CoAs. And again, tracking your carbons, each acetyl CoA has 2 carbons.
Yes.
Now, our goal is to take that acetyl CoA and extract all the energy out of it, all those high energy electrons out of it. We want to remove the electrons out of it. So removing electrons from it means we're doing what to it?
Oxidizing.
We're oxidizing. Leo, we love Leo Gur.
Losing electrons means you're oxidized.
This is a Leo Gur podcast here. Yes. We don't mess with oil. And so
oil rigs ruin the environment. Leo goes Gur. Everyone loves a lion. That's all we're saying.
Lion, right? We're extracting those electrons and then converting those carbons. Remember when last time we oxidized those carbons? We're turning it into CO2. We're turning it into CO2 and then reducing some other things, right? Getting those electrons, extracting them, giving them those high energy carriers for the electron transfer chain. Our goal is to extract the energy out of it during the TCA cycle.
There you go.
And so the entry point, like we said, PDC, the entry point was pyruvate output acetyl CoA. The entry point for the TCA or the Krebs cycle or the citric acid cycle is the acetyl CoA. Now, this acetyl CoA, this 2-carbon molecule, is going to combine with oxaloacetate. That's kind of the first kind of entry point, right? And actually, even before I start going the cycle, I need to give more context. I just realized I missed so much context.
I feel like you're diving in face first here.
Because I get so excited with it. We know acetyl CoA is the entry point, but we have to recall where did the PDC occur? Right? The PDC occurred in the mitochondrial matrix. The TCA cycle also occurs in the mitochondrial matrix, and that makes sense. The acetyl CoA is already there. Why would we do it someplace else? Right? So it occurs in the mitochondrial matrix. The acetyl CoA doesn't have to go anywhere. And as a result of that acetyl CoA going through the TCA cycle, there's some products that we produce. We already discussed that we're going to oxidize that acetyl CoA and turn it into CO2. So every time an acetyl CoA goes through, though, that 2-carbon molecule becomes 2 CO2. Well, we have to create those high energy electron carriers, like I was saying before. So for every single cycle that we do, for every 1 acetyl CoA that we do, 3 NADHs are produced, 1 FADH, 2 is produced. And so those are your high energy electron carriers. We took the electrons out of the acetyl CoA, we gave it to these 3 NADH and 1 FADH2, and we also produce 1 GTP. Right. And this kind of 1 GTP is kind of like a kind of a strange one, because we call it is GTP that is produced, but we kind of call it an ATP equivalent because that GTP can be converted to ATP. So whenever we talk about the citric acid cycle, we call this 1 GTP just kind of like an ATP equivalent. We kind of consider it as an ATP, even though it's not. But that's per cycle, per 1 acetyl CoA. But we have to recall a glucose, as a result, produced 2 acetyl CoA. So this happens twice. So per 2 acetyl CoA, that means we would produce 6 NADH, 2 FADH2, 2 GTP, and 4 CO2, right? The inputs and the outputs, okay? I don't think I missed anything when it comes to the big picture. I don't think so. I always kind of got too excited and jumped into it, but I think that's like the big picture.
That's pretty much it for the big picture. Yeah. And then recognizing that I don't think we verbalize this. It's a big circle.
It's a big circle.
Most of us have seen this before. We've seen the pictures, so we kind of know this intuitively, but it's a cycle, right? It's not a linear start line, it's not a race to get to the finish line. It's a lap, right? So every turn of that cycle is going to have those products. But what we're going to see is the end product of that cycle is going to be the reactant for the next round of that cycle. So just to track carbons, right? Acetyl CoA coming in as 2 carbons. It's going to fuse, like you said, with oxaloacetate, which is 4. And then we know every turn we're going to lose 2 CO2. So we have a 2-carbon molecule plus a 4-carbon molecule to create essentially the first reactant or intermediate of the citric acid cycle. And then we lose 2 CO2 in the process of coming back to that 4-carbon oxaloacetate. And then again, we fuse with the 2, take it back around, get back to 4, fuse with 2, lose 2, fuse with the, you see what I'm saying?
It's a cycle. It just is going in a circle always.
Right? We keep going around and around. And I think what's really important, because there's so many steps, right? And again, with glycolysis, we really dove in deep with pretty much not every step, but most of them, because you need to know them all. What I really think changed the game for me is recognizing that what you truly need to learn the TCA cycle and never need it again is really just one mnemonic. One mnemonic and maybe like 2 or 3 bullet points of knowledge, and that's it. So the mnemonic that I learned is inappropriate. So I will be giving you a
PG version.
A PG censored version, which is, can I keep selling shells for money, officer? So we've got our little shell stand at the beach. We're trying to sell it, and maybe we didn't have a permit to sell our shells. We're in trouble with the law for this.
Awful.
So, hey, I'm just, I live in Southern California. There's shell stands.
Of course. Yeah.
We're talking shells. This is the PG version. So that's the visual that you need, right? Our shell stand owner having this conversation. Can I keep selling shells for money, officer, please? Right? Not actually, please. That's not a word or that's not an intermediate, but those are each of the intermediates. And if you can remember them, we'll go through all the names in a moment. If you can remember the order and what the letter stands for, you can infer almost every single enzyme as far as what it is, what it does, and what its name is.
Super intentionally named. Yeah.
It's really nice. I feel like before we just list all the intermediates, I feel like we can do this all in one go. Maybe, Mike, let's go through reactant, product, enzyme name, next step.
Sure.
Right. We'll just
Let's do
go through
that.
them all because
Let's just,
it's
I
much
like
more
that.
topical than glycolysis.
Sure, let's do that.
Okay. I know you love the first step, so
I do
let's
love
go.
that first step,
I
right?
know
Because it
you
makes
do.
sense.
I stole
It
your
makes
thunder with it
sense.
a little bit. So
No, you're okay. No. Take the thunder. So like we were talking about before, the acetyl CoA is the entry point. And like we were saying, that's that 2-carbon molecule that fuses with kind of like the end point of the last cycle, which is the oxaloacetate, which is 4 carbons. Those combining together, fusing together, we do some wild math, 4 plus 2, and make our first intermediate.
7-carbon intermediate. Did I get that wrong?
Possibly.
Shucks, I lost my 100%.
maybe we can review that after, Molly. We'll chat about your math. Or you can come to the MCAT math session that we hold. No, fingers. So if you're better at math than Molly, you would realize that the first intermediate has 6 carbons, right? And that is our.
You're right.
You're so right. Yeah, well, MCAT math after. But the first intermediate is known as citrate. And this is actually why it's called the citric acid cycle. It's named for the first intermediate of the cycle, which is citrate.
Can I keep selling shells?
Oh, right.
More money officer. This is can. This is citric. This is citrate.
I was like, why are you saying can? Oh, the mnemonic. Right. Yes. And so, citrate corresponds with the first letter of our mnemonic, which is the can. And in can I keep selling shells for money officer. I also know the non-PG version. That's what always comes up in my mind.
Yes.
And so if we're thinking about we are making citrate, we're kind of like synthesizing citrate, you could say. So this is a real big hint. The enzyme that actually catalyzes this reaction, which is citrate synthase. Shocker, right? I love this because it's like it exactly what is happening. It's taking these two things and synthesizing citrate. Oh, of course it's citrate synthase.
Yes.
Did we memorize that? No, it just makes sense, right? And so this is when now we've kind of kicked off the TCA cycle. Also to note, and something that just came up that just kind of like really sparked my mind, a lot of people think that the PDC is part of the TCA cycle. And I'm like, yeah, I hear that all the time. Absolutely not. They're two separate things.
It's often shown in the same diagram as the problem. They'll have pyruvate coming in, acetyl CoA, and it'll say PDC on it, or Pyruvate Dehydrogenase Complex, and then it'll just feed right into TCA.
Yeah.
But they're different.
It's not part of the cycle. The first part is the citrate. It's acetyl CoA combined with oxaloacetate, being catalyzed by citrate synthase, and now you have the citrate. So now we've got citrate. We've started off the cycle. Take us through the next step, Molly.
Now we've got one that's a little less intuitive, but it's kind of fun. So we're going from citrate to isocitrate. What kind of reaction would you think that would be, going from citrate to isocitrate?
Well, I'm thinking the word iso reminds me of an isomer. So it sounds like an isomerase is doing an isomerization reaction.
Yes, which would be really nice if it was citrate or isomerase. But
would be amazing. It's not.
It would be nice.
Is the worst.
Yeah. This is one of our few weird enzymes. It's called aconitase. And it's actually named because there's there's an intermediate between citrate and isocitrate. So we technically go from citrate to aconitate, and then aconitate to isocitrate. There's an intermediate in this isomerization step. That is what it is named for. However, the same enzyme goes from citrate to aconitate and aconitate to isocitrate. So we ignore aconitate entirely because the same enzyme goes all the way from citrate to isocitrate. So that's how it gets its name. It is essentially isomerizing citrate into isocitrate, but its name comes from the intermediate formed as the halfway point between that isomerization. So the name is aconitase, but it's okay.
It's okay. All we know is nothing's really left or been added to the molecule, right?
Exactly. Nothing fancy's going on.
Yeah, we're just kind of reorganizing the molecule, getting it ready for the next step, which is the can I keep? So this is where stuff really starts happening, right? Because we're forming the next thing, which is the alpha ketoglutarate, right? And this is where, again, the action starts happening because now we're starting to extract some of the energy out of the molecule of that isocitrate. And by extracting energy, what we're saying is a redox reaction is occurring, right? We're taking that's what we're saying. Extracting energy is taking those electrons. Right? If I'm taking electrons, though, therefore something must be gaining electrons. If something's getting oxidized, something else must be reduced. Well, in this scenario, NADH is being produced because the NAD plus comes reduced, gains the electrons, and forms NADH. An important note here is that in this step, you also lose your first carbon, right? So we had a 6-carbon isocitrate. That's why, as a result of this step, you form alpha ketoglutarate, which only has 5 carbons, right? And in this case here, as a result, again, I just lost my train of thought for like a hot minute. And I'm like, oh.
I'm like, oh, he froze.
I'm like, froze. No, I just lost my train of thought. As a result of this reaction, this is a redox reaction. The name tells you what's going on. It is a dehydrogenase. But since we know it's a redox reaction, is it any surprise that's a dehydrogenase?
Nope.
No. And since the substrate was isocitrate, it's called isocitrate dehydrogenase, because we're doing a redox reaction with isocitrate to form alpha ketoglutarate. That's it, right? And so very intentionally named the can I keep redox reaction of isocitrate, removing some electrons, putting them on NAD plus to form NADH, but in this process, we also lose the first carbon to form CO2. So therefore, how many carbons, using that MCAT math we just talked about before, how many carbons does alpha ketoglutarate have?
12.
Well.
No, 5. 5
carbon. It's about 5 carbons. So this is kind of the first time where we're going down a carbon. So now we have this 5-carbon alpha ketoglutarate.
Yes. And the next step after that, so can I keep selling? We're going to do something totally wild, which is the exact same thing that we just did.
Amazing. Sick. I love that.
So we had alpha ketoglutarate, right, formed from isocitrate with isocitrate dehydrogenase. We gained an NADH and a CO2, right, or we lost it from the molecule. Alpha ketoglutarate to succinyl CoA is going to create an NADH and a CO2 using an alpha ketoglutarate dehydrogenase.
What a shocker.
I know, same naming scheme, same products. Although this reaction, we also bring in that CoA, right? Or the CoAsh. So we invest that in, and then we're basically about to chop it right back off.
Exactly, right. which is great. we kind of formed it, and then it's like, okay, we're about to take this away now.
Which is kind of similar to glycolysis.
Right, we add it, then we just chop it, which is cool. And again, name super intentional. So we just had 2 dehydrogenases. We've produced now at this point, we produced 2 CO2 and 2 NADH, right? Now we have this 4-carbon succinyl CoA. Now, I like thinking about this CoA. I don't even know if this is entirely correct. I think that this is a high in energy. Like, I like thinking this is a high energy CoA molecule. And so we're about to chop it off. That's what we're doing. We add the CoA. Now we're about to chop it off. But again, because it's high energy, I can couple that with something that maybe is not as favorable, because chopping off something that's high energy, that wants to happen. Something that's high energy doesn't like to be a case. It's unstable. We want to chop it off. But because it's very favorable, we can couple that, again, that keyword, to something that's maybe a little bit unfavorable, right? And this is where we have the step of coupling succinyl CoA cutting with the formation of that GTP that we just discussed, right? That I discussed at the beginning of today, of the TCA part, where we take GDP and an inorganic phosphate and we put them together to form GTP. Again, that phosphate does not want to be on the GDP. They're both negative. They don't like being together. But because we've coupled this unfavorable thing with a favorable cutting of the succinyl CoA and form succinate, kind of like succinyl CoA, just without the CoA. And this process is catalyzed by an enzyme that I think, again, can be a little bit misleading, right? In terms of in terms of naming, it's succinyl CoA synthetase. Now, the key here is, I think this is very similar to like the pyruvate kinase situation, where the name of the reaction is showing you the opposite of what's actually happening, right? In the pyruvate kinase situation with glycolysis, even though it's a kinase, it's taking off a phosphate and adding it to ATP. Even in this case, although it's succinyl CoA synthetase, it's actually taking apart succinyl CoA, not forming it, even though the backwards reaction would be forming succinyl CoA. But that's the idea, where it's not like an entirely random name. It's just the opposite reaction is how it's named for.
Yeah.
Mm-hmm.
So we've made succinate, right? And I want to be clear. Can I keep selling shells? Selling shells, there's two S's in a row. So just keep that in mind. Succinyl CoA is first, then it's succinate. Succinate is then going to be flipped into fumarate, right? I was going to say oxidized because it is.
Yeah, it is.
It's hard not to with succinate because succinate, this enzyme that catalyzes this reaction, is a really important one. It's called succinate dehydrogenase, which is exactly what you would expect because we have a catalyzed oxidation reaction from succinate to fumarate. Succinate dehydrogenase. Of course, if we're oxidizing succinate into fumarate, we are also making, or we're splitting off an electron. In this case, we're FAD to FADH2. You also will hear succinate dehydrogenase mentioned in the context of ETC, and that's because this is actually one of the complexes of the ETC. And I feel like a lot of people don't realize this because NADH is produced and then kind of floats over to the ETC. Succinate dehydrogenase is literally complex 2. Like it is complex 2. FAD that's in this reaction that creates FADH2. We don't have FADH2 float away from succinate dehydrogenase and go find complex 2 and donate its electrons. Yeah. It's literally FAD is part of the electron transport chain. It accepts those electrons from that reaction and then directly passes them into the electron transport chain. So this,
No, I was just going to say, like, that it's such an important part to recognize that, because I know some students are like, oh, there are so many dehydrogenases. How do I know which one's NADH, which one's FADH2? Well, there's only one that does FADH2, and that's the one that's in the electron transport chain, which we normally correspond with picking up those electrons from FADH2. So as long as you recognize it's the same enzyme, you don't actually have to memorize it at all. It's just the same one that's in both.
Yeah. Super cool. So that's the physical tether between the electron transport chain and the citric acid cycle.
Right.
They're both in the mitochondria, but that physically ties them.
And so now we've got, can I keep selling seashells for? So now we've just formed the fumarate. I also like to remember that fumarate's formed because FADH2 happened and you like pair the Fs. FADH2 is formed. Oh, you also formed fumarate when you formed the FADH2. A little kind of great memory piece there. But now, can I keep selling seashells for money? Now we're making the money, right? But this step, fumarate to malate, is a step that not that much happens. I'm going to be completely honest.
We're not making any money there.
Like we're not really making. We've got to be every step up since like the isocitrate. Something big's happened. Like we had a redox reaction, then another redox reaction, then we formed GTP, then we formed FADH2. All these things. Now fumarate to malate, we like add a water. That's it. Like we've just hydrated a double bond. And this is catalyzed by fumarase, right? Because it's fumarate is the substrate and it's fumarase. There's not much happening other than that, which I think is great, cool. Kind of a boring step, I'm going to be honest. But now we're at money, which is great.
Don't hate on the fumarase. Fumarase does its job just like everybody else. Just because it's not a flashy reaction, Mike, doesn't mean that it's boring.
I'm gonna say it's boring. Haters.
Oh my gosh!
Fumarase haters. Fumarase haters are on my side here. But hey, but now we formed that way. But in the next step, something more happens because now we have malate. So can I keep selling seashells for money? Officer. Right. And so during this step, this is where if you're keeping track of our carbons, our molecule now, that malate has how many carbons?
4
carbons, right? There's only 2 steps that remove the carbons. We've removed all the carbons we need. But now malate to oxaloacetate. Well, we recall the oxaloacetate originally had how many carbons?
4.
4. So we're going from a 4-carbon molecule to another 4-carbon molecule. So no carbons are being removed during this step, but we are seeing the formation of another redox reaction, right? We see another redox reaction here where we are removing we're getting that last bit of energy out. Because you might think, oh, all the carbons are gone. There's no more energy left. No, there's just a little bit left that we want to squeeze out. So we take those last electrons from malate to form oxaloacetate. Well, taking those electrons, well, that means we have to give those electrons to something else. And therefore, we add it to NAD plus to form NADH. But like I said, whenever you have a redox reaction, the name of the enzyme is malate dehydrogenase, right? Oh, a redox reaction, the enzyme's called the dehydrogenase. What a shocker. But it's malate dehydrogenase, where the substrate is the name of the enzyme. And now we form oxaloacetate. And look at this full circle. Look at this full circle formation where we started with oxaloacetate with acetyl CoA. We've gone through the whole cycle. We formed a bunch of NADH, FADH2, GTP, and now we're back with the oxaloacetate, which is ready to fuse with the next acetyl CoA and do it all over again.
Yes. So overall, if you know, can I keep selling shells for money, officer? You know all of it. The only enzymes that are weird in terms of their name is aconitase, which again does citrate to isocitrate. That one's weird, but again, name for that intermediate. The others pretty much all make sense until we get to succinyl CoA to succinate, which again, it's named for the reverse reaction. So it's succinyl CoA synthetase, even though that's what's being broken into succinate. It continues to make sense pretty much all the way through. Those are really the only two weird enzymes. And that's pretty much everything that we truly need to know. We could talk about regulation a little bit if we want.
Sure.
It's definitely tested more topically than glycolysis and less topically or more topically, so less deep than PDC. There's not really a ton to say here. It follows the same basic
principles.
Yeah. The only thing that's a little bit unique to this is that when we talk about things that that activate the pathway, we talk about things like ADP, for example. Again, the cell is in a lower energy state. We need to make more energy. ADP should upregulate the enzymes of the citric acid cycle. We also see calcium. And calcium is an important molecule in a lot of different ways. I think the easiest way is probably to think about muscles. If you haven't studied muscles yet, just know that calcium is intricately involved in the stimulation of muscle contraction. There's a lot that we could talk about, and I know that Mike loves my Romeo and Juliet story.
I can't wait. We need to
muscles.
figure out when we're going to talk about muscles, because I think that's so important.
Yeah, it is.
We'll talk about it another day.
But calcium plays a very important role in actually allowing for cross-bridging to occur in the muscle, which will shorten it, which allows for movement. And again, calcium can also be involved in other signaling cascades. Not important, but this is also something that will upregulate. And again, just think, if this is involved in the muscles, if I have a signal for a muscle to contract, that's going to require a lot of ATP. So having calcium around is going to be a signal to upregulate the TCA cycle as well. I think that's pretty much it. Maybe just also hitting on the specific enzymes that are regulated. Right. So citrate synthase is a big one. Isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Those are the big three. And it's
notice how fumarase wasn't part of that conversation. So
such a jerk. Really, it's kind of it's almost the first three enzymes if we ignore aconitase, which I wouldn't, but you would, because it's not
because it's kind of boring. So
so it's pretty much the first three big reactions are regulated steps.
Right. But that's really it when it comes to regulation. And this is where I think kind of always seeing the big picture, right, is so important, right? A lot of times we just try to memorize, like, oh, let me memorize which molecules are are regulating pathways. Oh, do I need to memorize every molecule? No. As long as you understand the big picture of the molecule, like what the goal is to get energy out, then it just makes sense why these molecules, regulate these steps, right? To not memorize them, I think, is the really big key, because it does just make sense.
I agree. And that's pretty much it as far as the content goes with PDC and TCA. We've had a lot that we've talked about. The only other thing that I want to mention is, so far we've given all of this material in the context of glucose metabolism, because again, that's really what's tested. And ultimately, we have gone from glucose now all the way to carbon dioxide. We haven't made water yet. We'll talk about that component next episode, actually. So far, we've taken glucose and we've made carbon dioxide and then also NADH and FADH2, along with some ATPs, right, for energy. But for the most part, that's what we've done so far. I want to acknowledge that there are other types of biomolecules that can be used for energy, and they actually are broken down initially by separate pathways, but they create intermediates that fit into this glucose catabolism pathway. And then from there on out, use all the same enzymes. So amino acids, we're not going to cover all the entry points because it's really not going to be super important for test day, but know that there are 2 different types of amino acids, 2 different classes. We say glucogenic amino acids that can be used to make glucose, which would mean logically, where in this cascade do we enter? Do we enter into some glycolytic intermediate? Do we enter into the PDC? Do we enter into the TCA cycle? If we make glucose.
If I make glucose, it has to be before the PDC. It has to be. Because again, if we're making it, acetyl CoA cannot go backwards to make the glucose. So it has to be at some point before that.
Yes. Yeah, absolutely critical. So PDC cannot go backwards and we don't have a different enzyme. So if you have a glucogenic amino acid that could be used to make glucose, it has to be broken down to some intermediate of glycolysis, because then it could just hop the reverse reaction of gluconeogenesis and make glucose. There are amino acids that can't do this. We call them ketogenic amino acids. And there really only are 2. So it's kind of
Like
a
purely
fun little
ketogenic.
fact.
Yeah.
There are only 2 that are exclusively ketogenic, and it's the L's, right? So leucine and lysine. And those cannot be used to make a glycolytic intermediate. All they can do is make some intermediate of the TCA cycle, right? Or they could be used to make acetyl CoA, but they cannot be used to make pyruvate. So that's kind of interesting. Again, there are enzymes that do both, that can be glucogenic or ketogenic, and then there's some that are glucogenic. But it's cool. Amino acids can enter this pathway. That's how you can metabolize proteins for energy as well. And then we could also take lipids, right? Especially triglycerides, which is a glycerol and 3 fatty acids, which is our energy storage form in terms of lipids. We could take those and metabolize. The glycerol backbone of a triglyceride can actually be converted to glucose. It can go in kind of with that glyceraldehyde can be converted into the glycolytic intermediate that way.
Great connection. Way from back. Yeah.
From last episode. And then the fatty acids can actually be broken down into acetyl CoA through a process called beta oxidation. So both amino acids and triglycerides can be used for energy. Some of those components can be used to make glucose. Others can only be used to make either fats by going through acetyl CoA or make ATP, right? By going all the way through to the end of TCA cycle, fueling those reduced electron carriers, and then ultimately we'll make that ATP when we go to the electron transport chain, which of course is where we're going.
Of course, which is going to be, if you haven't guessed it at this point in the episode, we're going through entire aerobic metabolism. If you've looked at big reveal. We've gone through glycolysis. We've gone through the PDC, the TCA cycle. Well, then naturally, next episode, we're going to be talking about the electron transfer chain and oxidative phosphorylation, which I know Molly's a huge fan of. I'm also a huge fan of it. Love it. You love especially when it comes to the redoxes, the oxidative, the reactive
and the connections.
I love the
just
coupling.
A bridge.
It's so cool. So I'm super excited to chat about it with you, Molly, because I think it's going to be really, really fun and great for everyone because, again, this is another area where people just trying to memorize these things, just trying to memorize, what are the complexes and what order are they, right? But no, we have to understand why. Why does the electron transport chain do what it does? There's so much meaning, so once you connect it, just makes so much sense. So when the passages ultimately start doing quests, I think the electron transport chain is one area where I see all the time in passages, they always love adding things or changing things up with the electron transport chain. Oh, we add this channel, what should happen? Oh, we inhibit this complex, what should happen? And so you can't answer those questions if you just memorize it. You need to understand it. So I'm very, very excited for next episode.
Yeah. And it's a good recap of a lot of the things that we talked about, which again is how you should be studying right? You should be looking for the connections to the other material you've learned so far. Don't just learn everything in its own little box and try and memorize it and shove it in your head. So we're going to talk about the connections to a lot of different stuff. Enzyme kinetics. We'll talk about redox. We'll talk about electrochemistry. We'll talk about different metabolic steps. We'll talk about a lot. Okay. So get excited. I know I'm excited and I know you are. And if you have not listened to some of our other episodes, please, this is a good time because we're going to see a lot of connections in the next one. So we hope that you enjoyed our episode today. Go check out our others. And we can't wait to see you in the next one. It's going to be lots of fun. So have a lovely rest of your day, week, whenever we caught you.
Whenever we catch you. And happy studying. I took it today. Yes, that's me. All right. See you later, everyone.
Bye.
Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash.